Okay, I hope everyone can hear me. Good afternoon and welcome to the Australian Stockholm Junior Water Prize finalist presentations. I'm Tomo Okai and I look after the National Awards program here at the Australian Water Association. A quick reminder that this session is being recorded and will be shared publicly with those who weren't able to join us today. I'd like to start today's webinar by acknowledging the traditional owners of the land in which we all meet today and pay my respects to elders past and present. I extend the respect to Aboriginal and Torres Strait Islanders joining us today. I'd like to also introduce the AWA board member and chair of the Member Experience Subcommittee, Sandra Hall, who will be facilitating the Q&A sessions today. The Australian Stockholm Junior Water Prize is the biggest water science competition for high school students and is proudly supported by Xylem. The prize aims to challenge young people to think big and take a fresh look at local and global water problems. The winner announced today will go on to represent Australia on the international stage, competing with over 35 countries later this year in August. It is truly inspiring to see the dedication and hard work, and we'd like to commend them for producing such high quality, thoughtful solutions to water quality issues. Today, you'll hear from 4 innovative young water scientists: Max Zhao from Barker College, Julia Cummins from PLC Sydney, Miriam Gray from Cooks Hill Campus, and Sisti Zhao from Queensland Academy for Health Sciences. Before we proceed any further, I would like to invite from our award sponsor, Xylem, the Managing Director, Australia and New Zealand, Brian Krishna, to say a few words. Welcome, Brian.
Thank you, Tomo, and welcome everyone, and thank you for having me here today. I'm really excited to be here for today's proceedings. Well, we are delighted, of course, in association with the AWA to sponsor the Australian Stockholm Junior Water Prize. And in fact, Xylem is also proud to be the founding global sponsor of the Stockholm Junior Water Prize since 1997. Encouraging youth in our communities to bring in new ideas, your energy and passion to solve the world's toughest challenge. And the reason we at Xylem exist, our vision is to help create a world in which water issues are no longer a barrier to human health, prosperity and sustainable development. In a world that we've seen evolve so much over the past 100 years, I often ponder why is it that so many have still been left behind in the want for what should be a basic human right to have access to clean water and sanitation. Since industrialisation, we have seen more water used in everything that we have today. Manufacturing technology advancement has become more water intensive. However, being responsible for water is not always the industry's first thought when it comes to making new products. It is so important that we at Xylem continue to invest research and development, improving water technology to help all industries and communities be aware of smarter ways to use water. It's our job and it's your job to be the next generation that can provide these innovations on smarter ways of using water more efficiently and effectively, and then returning it back to the earth in a better way than we found it for the generation still yet to come. It's important that you share your stories, not only through some of the great ideas or community engagement that has brought you here this week, but in general about water, knowing how did it get here, how much do we have, how can we use it better. These are many questions that our populations, both locally and abroad around the world, need to continually be educated on. Finally, I thank you for being here. We're really excited about the future of water with you in it, and we really look forward to today's presentations. Thank you.
Thank you, Brian and Xylem, for your generous support. The judging has all been completed prior to today, so this webinar is really about showcasing these incredible young minds. Each finalist has been given 8 minutes to present their project, which will be directly followed by 4 minutes of question time. If you do have a question, please type it into the Q&A box at any point during the finalist presentation. I'd now like to hand over to Sandra to introduce the first speaker for today. Thank you, Sandra.
Good morning, everyone, and thank you to Tomo and, of course, our sponsor for this prize, Xylem, and Brian for his words before. Look, I'm really excited about this. The Stockholm Junior Water Prize is always one of the highlights for me as chair of the Honours and Awards for the association. I'm really inspired by the young people and interested in who inspires them. So our first presenter today is Max Zhao. Now, Max was inspired and was encouraged by his tech teacher at school who submitted to the young scientist and then progressed his project to go into here. And Max from Barker College is now studying data science, one of the subjects and courses that I am really interested in at UNSW. And he'll be presenting his project this afternoon on remote control raft. So welcome, Max.
Hello, can everyone hear me?
Yep, all good.
All right, perfect. So welcome everybody. Today I'll be running through the process as to how I developed my project over the previous year. So we'll start right at the very beginning with the problem that I chose to address. So 29 million metric tons of plastic waste enter waterways globally, harming ecosystems, creating health hazards, and accelerating global warming. Plastic waste threatens over 3,000 marine river species, including birds, fish, crustaceans, reptiles, and mammals. Large pieces of plastic, including bags and bottles, become floating snares and can suffocate and choke animals that encounter it. However, it is the microplastics that pose the largest threat. When fish consume plastics, they die younger due to poor nutrition, and this is then passed on to any species that consumes the fish, including people. In America alone, 70,000 people are hospitalized from eating plastic-contaminated fish. Due to microplastic pollution within freshwater bodies, tap water within certain areas of China have become, to a certain extent, undrinkable. Many people have become hospitalized, then paying for excessive medical fees. So in recognition of the risks, millions of households purchase bottled water, which is not only extremely costly in the long run, but also further contributes to the plastic problem. Furthermore, studies show that plankton are consuming more microplastics than ever before, which will aid in the rapid process of global warming. Previously, oceans have absorbed 30 to 50% of carbon dioxide emissions from human activities. However, as plankton consume more microplastics, their microalgae growth is significantly impaired, reducing the efficiency of photosynthesis. Hence, their ability to remove carbon dioxide from the atmosphere is decreased. So, my project seeks to resolve the problem of floating marine waste on a smaller scale and in a more targeted manner. The term larger scale would refer to projects like that of System 001 and Interceptor 002, developed by the company Ocean Cleanup. System 001 is an enormous vessel that spans 1 kilometer long, deployed in the Pacific Ocean, that gathers waste by using the current as a method of collecting waste in a huge net, sort of like a filter. And Interceptor 002 is a custom boat-like vessel that operates in numerous rivers across the world, collecting waste using a large conveyor belt. So then, by the term smaller scale, I'm referring to local efforts to control marine waste in rivers, lakes, and essentially any place where land meets shore. So activities taken to clean up areas on a small scale are predominantly manual tasks whereby volunteers must have to net waste from shore, and this can be extremely laborious and time consuming, requiring immense manpower to yield any significant impact on the environment. So my project is a small and compact aquatic raft designed to collect floating plastic waste. It is remote controlled so that a person on shore can maneuver it and activate the plastic intake mechanism from shore. The main purpose of my project is to provide a more targeted and thorough cleanup of natural waters such as rivers and lakes by accessing areas where humans previously couldn't, such as underneath jetties or underneath bridges. So this way, communities and local environmentalists can enact a more thorough cleanup of waterways without putting in as much time and manpower that would otherwise be required. So first of all, there were 5 main factors for consideration before finalizing my design. So firstly, movement, then user interface, power, size, and then waste collection. So when approaching these factors, I had to find the most effective way to execute them within the given time and resource constraints. So for movement, I chose to use propellers as a method of propulsion, which will be comprised of a dual motor system. So this will allow for a greater right and left turning ability compared to the rudder, as it has a larger turning radius. For user interface, I decided that the simplest and most effective option was for the raft to be controlled remotely through Bluetooth connection. So this increases the accessibility of the product's use, as most people have an Android or Apple phone that can easily be connected using Bluetooth. As for power, although I would have liked to use solar power, which would have been the most desirable method due to its sustainability, it was not viable due to the amount of time and effort it would take to implement and integrate into the system. So, and then for the factor of size, my raft is directed towards small scale cleanup, and within my brief, it should be able to access small cramped areas where humans cannot enter, so it will be small, compact, and portable. This is a larger factor that differentiates from other solutions. And as for waste collection, my waste collection will be limited to floating wastes, as underwater collection is vastly more complex. The conveyor belt and suction method require more power than the roller method, which I'll go onto later in my slideshow. So to test my idea first, I constructed a test proof of concept for the RC raft cleaner, utilizing PVC pipes, and two large PVC pipes served as pontoons upon which the floating intake could be activated. It was not designed to float on water, as I hadn't developed any waterproofing mechanisms, so it was primarily used as a way of testing the configuration of the entire device. Since at the time I only had one waterproof 12-volt motor, I used small DC motors in place of the propeller motors. And from constructing this test proof of concept, I determined that the project was viable, but it revealed many areas for development, including waterproofing, cable management, flotation, and the design in general. So after committing to this project, I began researching the main aspects of it. And my research was divided into two main categories: the system it would operate on and the physical design. So under the operating system, I researched appropriate programming languages, microprocessors, and motor options, which would then allow me to construct the core circuit. So for the programming language, I chose C++, as both my parents use it, and they could be an immediate source of help. Also, it's the most powerful language and is also widely supported, making it very easy to research online. Next was the microprocessor. So of the three shortlisted options, which I had as Arduino, Raspberry Pi, and the BeagleBone Blackboard, I chose Arduino as the most befitted to the purpose of my project. I have the most experience with using it out of any of the other boards, and it's also inexpensive. And its features are mostly catered to the system I want to create for remote control and motor control. As for intake, as you can see on the right, there are three of my initial ideas for intake methods. The first one is a pump and filter, where it creates an underwater vacuum that essentially creates a directional flow of water. So then as the water flows through, the raft, acting as filter, collects any waste that is on the water surface. The third is a conveyor belt that carries the waste over the water surface and into a storage area. However, it was the second design that I selected with the roller mechanism. The first two designs that I mentioned would not have been feasible within the given time and resource constraints. As for the raft design, I chose a bow and hole design. My project is not designed for speed, but rather for navigation and proper positioning for cleanup. So my design should promote stability over agility, and that's why I made the selections I did. And as for production methods, most of the 3D printed parts, most of them are 3D printed, since I'm proficient in CAD, and this would be the best way to produce custom parts. And for motor options, I chose 7.4 volt waterproof motors, which would befit the circuit. So here is the core circuit in the top left corner. As you can see, there is a diagram of the circuit which uses an L298N motor driver to control the motors, and a HC05 Bluetooth module that can connect to any mobile device. Before implementing the circuit, I replaced the motor configurations with LEDs. So each of the different colored LEDs correspond with a different type of movement, represented in the code on the bottom left. So on the bottom right, I connected the circuit to an Android phone and displayed how different inputs created different movements as depicted with the different combinations of LEDs. So here is my final design from the sketch I created in CAD. So I created a CAD model that best embodied the sketch I made on pencil, and this would be very helpful to me later on, allowing me to 3D print the parts I needed. And at the front of the device is a roller mechanism that put the debris into the net behind it, as you can see. And this last slide is just a compilation of the final tests and constructions that led to the final product. So using the Arduino Bluetooth controller application, I was able to utilize the joystick function to create an intuitive and easily accessible user interface for people to use. Then by printing a small housing structure, I was able to move the electronics from the breadboard into a very compact space that would fit inside the raft. Also ran tests on the raft's ability to float and the most effective roller designs, which ultimately led me to the final product. So once constructed, I tested movement and intake ability, to which were both successful. And on the bottom, you can see I put a ball inside a pool and then tested its ability to move around that ball. So if there are any questions now.
Thank you very much for that, Max. I loved the kind of designs that you tried out, and you already thought about the support and the collaborators and your parents with the CPAS. So loved it. Now we do have one question here from Maria. What is the approximate amount of plastic that your device can recover from water in, say, an hour?
Oh, yes. I forgot to mention in the previous slide that I also did tests on how much weight it could hold before it would eventually sink and the electronics would be. So I tested different weights, and the maximum holding capacity right now is 5 kilograms.
5 kilograms. Great. Oh, thank you for that answer. Doesn't seem like very much plastic, does it? But when you talked about the extent of the problem, so maybe we need to think about a larger design.
Yeah.
Yeah. Awesome. Thank you very much. Max. Now we're running out of time, so we're going to move on to the next presenter.
All right. Thank you very much.
Thanks. Our next finalist today is Julia Cummins from PRC Sydney. And Julia had a similar transition into this award from the Young Scientist Award, and encouraged by her teachers. Julia is now studying medicine at Western Sydney University. And today she'll present her project, Why Silver Might Not Be Gold in Water. Julia, welcome.
Thank you, Sandra. So good afternoon, everyone. Today I'll be presenting my project, Why Silver Might Not Be Gold in Water, an investigation to determine the minimum concentration of nanosilver which causes toxicity to Daphnia magna. If you've ever brushed your teeth, worn makeup, or used an electric shaver, chances are that you have been in contact with nanosilver. Nanosilver, as shown in Figure 1, are nanoparticles of silver 1 to 100 nanometers in size. Nanosilver has strong antimicrobial properties, leading to it being used in clothes, cosmetics, textiles, and wound dressings, as shown in Figure 2. However, products containing nanosilver release it into the environment from industry and consumer products. The goal of this project is to determine what concentration of nanosilver is not damaging to ecosystems. Although silver has been safely used as a biocide for centuries, nanosilver is more toxic than bulk forms of silver, as its smaller particles means that it has a higher surface area to volume ratio. This accelerates the dissolution and release of silver ions, the primary mode of nanosilver toxicity. As shown in Figure 3, silver ions have the capacity to penetrate cell membranes and cell walls, entering cells to then damage cellular structures and molecules such as proteins, lipids, and DNA. This eventually leads to cellular death. Nanosilver has been proven as toxic to the aquatic organism Daphnia magna, so it's therefore considered a pollutant. To mitigate the damage of pollutants in the environment, threshold levels are developed. Threshold levels are values under which a healthy marine ecosystem is present. While there is a well-established threshold level for silver set by the New South Wales Environmental Protection Agency, there is no such threshold level for nanosilver, as despite their different particle sizes, nanosilver and silver have different molecular properties. And this means that currently they are subjected to the same reporting requirements, despite the abundant scientific literature that indicates that nanosilver is more toxic than bulk forms of silver. It is currently, therefore, unknown what the threshold level for nanosilver is, leading to the aim to determine under which threshold level nanosilver does not exhibit toxicity in the aquatic organism Daphnia magna. To investigate the aim, a bioassay will be conducted using Daphnia magna, small planktonic crustaceans as pictured in Figure 5. Their population and heart rates will be monitored in different concentrations of nanosilver.
There are a number of hypotheses for the population and heart rates as shown on the screen. The null hypotheses state that there is no difference between the tested concentrations, and the alternative hypothesis state that there is a difference. For this experiment, the main risks were glassware breaking and causing cuts, a slipping hazard presented by the liquid spilling, and human consumption of nanosilver, and inhaling nanoparticles from the air. Strategies to minimize these hazards include wearing safety goggles, gloves, a coat, and a face mask, and exercising caution when handling the equipment. To complete the project, the items from the equipment lists shown on the screen were collected. The equipment was then set up according to methods A, B, C, and D. Firstly, method A was the preparation of the nanosilver solutions. In 5 containers, the solutions were prepared according to figure 7. A note that colloidal silver is nanosilver only suspended in water. This was repeated so that there are 3 replicates for each of the different treatments. Figure 8 depicts a label diagram of the experimental setup. Each individual vessel contains 200 milliliters of nanosilver solution, with each containing 0 milligrams per liter, 0.25 milligrams, 0.5 milligrams per liter, 0.75, and 1 milligram per liter of nanosilver. Method B was the preparation of the Daphnia magna cultures. Figure 9 demonstrates how the lids of the 3 cultures were removed for aeration and then left for 24 hours. As we can see now in figure 10, a 3 milliliter Parsley pipette with the end removed at a 45-degree angle was used to place 10 individual Daphnia magna into each of the 15 total nanosilver solutions. The next method was method C, calculating the population of the Daphnia magna. Firstly, bird's eye view videos of the 15 containers were taken using a digital camera. From the photos, the dead Daphnia magna in the containers were then counted and recorded. As you can see in figure 11, the red circles denote the dead Daphnia magna and the green circles represent the alive organisms. Using the stopwatch, the population of the Daphnia magna in the 15 solutions was counted once every 15 minutes for an hour. Finally, method D was calculating the heart rates of the Daphnia magna. As shown in figure 12, a Daphnia magna was removed from a solution using a pipette. It was then placed in the well on a concavity slide under a digital microscope. On the digital screen, an image which is shown here, the heart was located behind the eye spot. Using the digital microscope's recording function, the heartbeat was then recorded for 5 seconds and watched later in slow-mo. Using the stopwatch, the population of the Daphnia magna was recorded every 15 minutes for an hour. So this is a short video of what I obtained under the digital microscope. So this is what I was able to see. So this is a close-up under 40 times magnification of the Daphnia magna organisms. So you can see the heart beating just under the anterior dorsal surface. And then this is the eye spot here, which was used as a point to locate the heart just behind. And so this is what I was able to see. And therefore, as you can see, because the heart's beating quite quickly, I had to record the heartbeat for 5 seconds and then watch it later in slow-mo to make sure I got the accurate amount of heartbeats. To adhere to ethical conduct in animal research as outlined in the Australian Code, this project fulfilled 2 standards. Firstly, the animals were suited to the purpose of the project. Daphnia magna are appropriate to use in this project because they are bioindicators, which indicate toxicity for other organisms. They are also sensitive to changes in water chemistry and are invertebrates without a central nervous system, so cannot therefore feel pain. Secondly, respect for the animals underpinned all actions involving their care and use. For example, the Daphnia magna were given 24 hours to recover from shipping shock before being used in the experiment. A relationship between nanosilver concentrations and the population and heart rates of Daphnia magna was observed, as can be shown in the clear trend in graph A.1 and B.1. For both variables, an ANOVA test showed that there was a significant difference between the treatments, as the p-value calculated for each of the treatments was below the significance level of 0.05. This provides evidence against the null hypothesis that there's no difference between the treatments. So looking at graph 8.1, the average population of the Daphnia magna, you can see that in the control, which is this red line up the top, the population did not change. And then as the concentration increases, you can see that the number of Daphnia magna living at the end of the experiment decreased quite significantly. This trend is also followed in graph B.1, the average heart rate of the Daphnia magna over the time period. So you can see that there was a decrease, most significantly noted in the nanosilver solutions with the highest concentration. Post hoc analysis of the population and heart rates of the Daphnia magna using the Tukey test found that the minimum concentration of nanosilver that causes toxicity to Daphnia magna lies within the range of 0.26 to 0.5 milligrams per litre. Thus, as nanosilver releases into the environment continue, to prevent potential toxicity occurring in living organisms, nanosilver solution concentrations in the environment must be monitored so they do not exceed the specific threshold level identified for nanosilver of 0.26 to 0.5 milligrams per litre. Thank you very much for coming today. I would now like to open up the floor, or should I say Zoom, to my audience with some questions.
Thank you very much for that, Julia. I'm blown away by how thorough you are, even going into the safety of the experiment. As a fellow scientist, I love that. Any questions from the floor? I guess I have a question. The Daphnia magma, if we were to use it as a long-term bioindicator of toxicity levels in water, how easy are they to find in the water body?
So you mean to obtain them?
Yeah. So if I was to go down to the local dam and test, could I find them?
In freshwater sources, yes, you could. So they are freshwater organisms, so therefore you have to locate a freshwater environment. They're not so much found in Australia. They're typically imported from North America, where there's a lot of more freshwater bodies, and that's where they're obtained. And then they're often brought here and used as bioindicators scientifically, or also as fish food. So people actually breed them as fish food. And so that's their other purpose outside of the scientific realm.
Yeah, right. And do you think there are other factors that could influence that toxicity level? Is there something that, if you were to expand on the project, what other factors do you think that you would test?
Well, obviously, most importantly would be the concentration of the nanosilver, which is the main factor that determines toxicity. But there are a couple of other factors that I would like to explore in future projects. So, for example, it could be investigated if the age or the gender of the Daphnia magna impacts how nanosilver affects them, because there's actually quite a significant size difference between male and female organisms, and their lifespan as well can be affected. And so I'd be interested to see maybe if the size of the organism or what gender therefore affects how nanosilver affects them. And another really interesting factor would be this experiment was undertaken in freshwater, but I'd also like to conduct it in seawater, which is where a lot of nanosilver emissions would also be entering the environment. So conducting it with different factors in order to determine exactly what the nanosilver threshold level would be.
Yeah, wow. Thank you very, very much, Julia, for your presentation today. If there's no further questions, we'll move on. I'd now like to invite our next finalist, Miriam Gray from Cooks Hill campus. Miriam did a bit of a project or some work experience with Hunter Water, who encouraged her to apply for this award. Miriam's in grade 11 this year, and we wish her all the best for her senior years. Today, she'll be presenting her project, How Does Grey Water Affect Plant Growth? Welcome, Miriam.
Thank you, Sandra. So thank you, everyone, for taking time out of your day to hear me discuss my Australian Stockholm Junior Water Prize entrance, which studied grey water's effect on household plant growth. All photos in the presentation are stock images, and I want to thank you for bearing with me as I've recently had COVID-19, so may exhibit symptoms during the presentation. I've decided to talk about four main things today. The first is an overview of my project. Then I'll go deeper into my results. I'll outline areas in which I believe further study should be conducted, and express my views on what I think should be done to increase the reuse of grey water into the future. So in my initial video pitch, which I believe is online for all those that want to see it, I discussed how much grey water the average person uses, the plants and irrigation fluids I used, and the future benefits to using grey water on the garden. I chose this project because of the drought in 2019 to 2020. In Australia, people were encouraged to implement water-saving strategies. One of these, which caught my attention, was capturing water used when washing vegetables or waiting for hot water to come through, and reusing this water on household plants. I wondered if this clean water could be supplemented with laundry grey water, and if it could, I believe this would greatly help relieve water stress on town and private water supplies. Previous studies show that grey water investigations have been performed on silver beet and tomatoes, among other plants. I chose 4 different plants: green beans, snapdragons, basil and parsley, and 5 different irrigation fluids to test. The control for my project was tap water, as this is what most people use on their gardens. I grew my plants hydroponically, so also included hydroponic solution as one of my fluids. My irrigation solutions were grey water made from earth detergent, grey water made from, I'll start that sentence again. My grey water solutions were grey water made from earth detergent, a grey water safe detergent, grey water made from cold power detergent, a non-grey water safe solution, and a diluted form of the non-grey water safe solution. I prepared the solutions by mixing the detergent and the tap water. So I've discussed grey water safe and non-grey water safe, but what do these terms actually mean? In laundry liquid and powder, there are a few ingredients that can harm plants and gardens. These components, as found in a study published by Choice, are phosphorus, salinity, and pH. While small amounts of phosphorus can be useful for plants, excessive amounts when it gets into our waterways can cause algae growth. All laundry detergents contain salts, and these are detrimental to plants and soils. Laundry detergents are also highly alkaline. The difference in grey water and non-grey water safe is that those deemed grey water safe generally have lower quantities of phosphorus and salt and have a pH that is more suited to biological growth. On to my results. To begin, green beans. The growth of the beans was supportive of non-grey water safe water. The plant in hydroponic solution was the tallest at the end of the investigation period on day 35, but this was expected as hydroponic solution is designed to give plants the exact nutrients they require for growth. However, this plant did not yield the most growth. That achievement was taken by the bean in the non-grey water safe liquid. There was minimal difference between the end heights of the various plants in grey water. This was due to the original heights varying slightly. This still gives supportive results for grey water being used on green beans. All snapdragons grew. The snapdragon in hydroponic solution produced the most growth. This is not surprising due to the nutrients that hydroponic solution provides. There was minimal difference between the final heights of the various grey waters and tap water. The plant in diluted grey water fared better than the others. These results support grey water being used on snapdragons. While grey water doesn't produce taller snapdragons than tap water, it still sustains life. The basil plants did not exhibit results in the same way that the beans and the snapdragons did. The plants survived equally well until day 24, when the plant in hydroponic solution grew exponentially. None of the fluids killed their plants. Further studies should be done on basil to determine more conclusive results. But from this experiment, it can be gathered that grey water, either deemed safe or not, still permits life for basil. The results from parsley were inconclusive at best and incorrect and misleading at worst. Fauna discovered the parsley plants on about day 7 on the 3rd of the 12th, I think that slide says. This fauna ate, pulled, and generally damaged the parsley plants. It's curious to note that the same fauna did not destroy the other experiments as they were in the same approximate location. Further study is needed if results about how parsley survives in grey water are needed in the future. Further studies should be conducted prior to laundry grey water being widely used on plants. Some possible topics that I've discovered include the following. The effects of untreated laundry grey water on the soil in the long term should be discovered. These negative effects need to be quantified before it is decided whether or not grey water can be used on plants. Studies should also be done to find out how grey water affects the soil in regard to harmful bacteria. If extra bacteria is placed in the soil from greywater, will plants increase their intake of this bacteria, resulting in a greater quantity of human consumption of the harmful bacteria? If human health is diminished by using greywater on edible plants, it's not a viable option for the future. To minimise possible long-term effects on the soil and any effects to human health, are there some simple, accessible and environmentally friendly greywater treatments that could be implemented by the consumer to reduce or eliminate these effects? Provided laundry greywater is safe for the environment, studies should be done to discover which plants greywater can be used on without detriment to the plant health. I also discovered that over half of a person's daily 100 litres of greywater comes from their shower, or more if they choose to bathe. Further studies should be done to determine whether or not this water can be used on plants, as it contains different chemicals and matter to laundry greywater. So how can we continue improving our stewardship of the water that we receive? If further study is conducted and greywater has been found to be safe to use without detriment to plants, the environment, or human health, legislation should be changed to allow household greywater use on gardens. This should be coupled with government subsidies to reduce the cost of installing systems to harvest greywater. Currently, systems range from $800 to $4,000 and even up to $20,000 for water treatment systems in Australia. This price is not accessible for most people. Greywater safe detergent should also be reduced in price so that it's more accessible for people to use. Some households can only afford the cheapest laundry detergent, and the cheapest is not normally the best for the environment. If the government can make the detergent that is best for the environment the cheapest, people will use it more. Thank you again for listening to my project and considering. I greatly appreciate this opportunity. I believe there's time for questions.
There certainly is. And Miriam, thank you very much for your presentation. I love that you've thought about what's next and all the questions that I had going through that presentation. You answered them all at the end there with what would we do next. So we do have a question from Maria Luisa for you. Greywater can also contain microplastics from the laundry, and Nanosilver as pointed out by Julia. How would you suggest this could be managed or minimised to be able to safely use the greywater for plant irrigation?
Yeah, so I think that the microplastics and the nanosilver and any other detrimental things in the greywater need to be discovered and quantified. Like if they exist, but in such a small manner that it doesn't matter, then we may not need ways to deal with the pollution. But if they are in large enough levels to cause a long-term effect, we need to discover simple treatments, like with the point of simple, accessible, environmentally treatments, both to help with the pH and salinity and stuff that I already discovered, and also those microplastics and nanosilver that Maria pointed out.
Yeah, thank you.
So further study.
Further studies. Oh, there's so many different ways you could take this project, and even you know, you obviously had a possum that potentially got into your experiment and ate your parsley. They're very selective on what plants they do eat. And so would you design the experiment differently next time?
Probably. I'd probably set it up in a way that fauna couldn't discover it so that I'd be able to get results. I'd also probably try different plants. So I've got results from beans and snapdragons. I'd try something else that hasn't been studied yet as well. Yeah.
Excellent. Well, if there's no further questions, I'll say thank you very much, Miriam.
Thank you.
And we'll move on to our last presenter for today. Our last finalist is Cici Zhao. So Cici is, again, it was a teacher that inspired her to apply for this award. Cici is currently studying biomedical science in Melbourne. Cici did this work while she was at the Queensland Academy for Health Sciences, and today she'll be presenting her projects, probiotic water treatment, the effect of probiotic lactobacillus strain on the inhibition and degradation of E. coli biofilms as a novel method for waterborne pathogenesis control. Now, I swear you put in those big words just to stump me this afternoon. Cece, welcome.
Thank you so much, Sandra. Okay. Okay, good afternoon, everyone. My name is Cece Jo, and I'm a recent high school graduate of the Queensland Academy for Health Sciences. My research is related to the inhibition and degradation of biofilms in water systems by probiotic microorganisms as a novel method for water pathogenesis control. I chose to use probiotics as I wanted to explore further the concept of bacterial competition and how that can be used to our advantage in fighting pathogens. In the light of growing antibiotic resistance amongst pathogenic bacteria, this possibility is increasingly significant. Currently, probiotics are most widely used to maintain gut health. They are known to be able to balance the microbiota by suppressing harmful organisms through secretion of antimicrobial substances. Bacteria exist in two forms in water systems, and that's planktonic, which is free-floating bacteria, and biofilms. Due to the structure of biofilms, they are very hard to remove. In developing countries, waterborne bacterial infections account for 80% of all illnesses. Water treatment methods such as chlorination and UV disinfection are shown to have adverse health effects or are economically inviable for large-scale implementation for developing countries. Detection of biofilm growth in the pipes of water distribution systems in developed countries has also been an ongoing problem, leading to infections as a result of water contamination. Thus, through my experiment, I wanted to test how well probiotics were able to suppress the growth of bacteria in a water system setting in their planktonic and biofilm state, measuring inhibition and degradation respectively. And using two different strains, I also wanted to discover whether a single species probiotic strain will be more effective as compared to a strain with multiple species. I chose lactobacillus strains to be used in the experiment as they are commonly used in dietary supplements and thus can be easily sourced. I sourced the single strain L. casei shirota from Yakult and the multi-species strain which contained a mixture of L. paracasei and L. plantarum from Swiss Probiotics. In the first step of my experiment, I isolated the cell-free culture supernatant, or the CFCS, of the activated probiotics through centrifugation. The CFCS was expected to contain the antimicrobial substances that probiotic organisms secrete. And 8 two-fold serial dilutions were conducted with the CFCS for each plate to give me a wide range of CFCS concentrations to work with. To measure the inhibitory effects, the serial dilution was conducted in the 96-well plate. The E. coli was co-incubated with the CFCS concentrations. And the plate was then incubated for 24 hours to allow any biofilms to form, and a crystal violet assay was used to quantify biofilm growth. To measure how well the probiotic CFCS was able to degrade mature biofilms, E. coli was first incubated in the 96-well plate for 24 hours. And again, a two-fold serial dilution was carried out externally with the CFCS in culture tubes. The CFCS concentrations were then inoculated into the wells, and the plate was then incubated for another 24 hours. Again, biofilm growth was quantified by a crystal violet assay and UV spectrometry. During the crystal violet staining process, we could see that the biofilms grew in a ring like you would see in a pipe. And furthermore, in the inhibition plates for both single and multi-species strain, it could be seen with the naked eye that there was a concentration group that had much darker stains than the rest. This trend can also be seen through the significant peak in the absorbance graph. The minimum inhibitory concentration was defined as the lowest concentration of CFCS where biofilm growth was lower than the control. This happened to be 25% for both strains. Overall, the single species strain showed lower amounts of biofilm growth and thus better inhibition. However, the multi-species strain was able to inhibit biofilm growth by 70%, while the single species strain only inhibited by 20% as compared to the control. For the degradation plates, the results were much more randomized. It can be seen for the multi-species CFCS, there's an increase in biofilm growth as the CFCS concentration increases, while the opposite occurs for the single species CFCS. The single species strain had no significant difference in absorbance to the control at CFCS concentrations of 1% to 25%, but was effectively able to degrade at 100% concentration. At 50% concentration, the degradation was not significant due to the large error bars. So the peak seen in the inhibition graph may be as a result of, firstly, the bacterial life cycle of lag, log, stationary, and death phases. The lower concentration could signify cells that are already dying, with cells at 1% and 2% well into the death phases, explaining why there's a decrease in absorbance from 2% to 1%, while the peak shows them at a log or stationary phase, where bacterial growth is most abundant, signifying that the CFCS was able to inhibit growth for a while, but was not concentrated enough. Importantly, the higher concentrations did effectively inhibit biofilm growth, at least for the 24-hour timeframe provided. A second explanation for this could be symbiosis and bacterial cooperation. As we are not exactly sure what is present within the CFCS, this may include the probiotic microorganisms themselves. The K12 strain of the E. coli bacteria was used, and in some instances, this strain of bacteria is considered probiotic. Studies have shown that lactobacillus, when incubated with probiotic E. coli, demonstrated promotion of biofilm growth due to bacterial cooperation. This is due to them occupying adjacent ecological niches, and thus there's no threat of direct competition. This explanation could also explain other occurrences of promotion in the other trials. A limitation of the crystal violet assay is that lactobacillus could have formed part of the biofilm during bacterial cooperation, but however, there was no way to differentiate between the species. Furthermore, more effective inhibition with the multi-species strain can be explained by synergistic properties as expected with the multiple probiotic species. However, overall, the single species strain showed overall lower biofilm growth when measuring inhibition, and it was a single species strain that was able to effectively degrade at 100% concentration. However, it is important to note that the L. casei-shirota was sourced from Yakult, which is a fermented milk drink, and thus the CFCS could have contained substances such as sugars that will be more beneficial to the survival of lactobacillus if they were present within the CFCS. Some extensions could include conducting this experiment in a PC2 laboratory and being able to work with pathogenic bacteria and studying their characteristics and how they might work with lactobacillus and probiotics, as well as modeling pipe geometry and setting and transferring this further into the real world. And of course, conducting mass spectrometry for the CFCS and identifying the individual components that may affect the growth or inhibition of biofilms. So from the research, it can be concluded that at high concentrations of Lactobacillus culture supernatant, there is successful inhibition of biofilm growth for both single and multi-species strain. The single strain culture supernatant was effectively able to degrade mature E. coli biofilms at high concentrations. And finally, using probiotics for biofilm control must be done carefully and selectively used against specific pathogenic bacteria that occupy the same ecological niche, so there is direct competition. These results can be utilized as the starting point on the usage of probiotics to inhibit and degrade pathogenic biofilms in waterways as a safe, effective, and economic method for biofilm and water pathogenesis control. Further experimentation can be conducted in order to continue transferring this strategy into the real world. Thank you very much.
Thank you very much, Cece. My undergraduate degree was in microbiology, and I loved all the graphs on growth. We do have a question from Maria. I want to thank Maria for asking all these great questions today. Thank you. How did you control the pH in your assays, Cece? Is your cult acidic? Could this have affected your results?
Yes, so actually pH was one of the many considerations that I put into while researching probiotics. So I knew that lactic acid was a substance that lactobacillus organisms secrete. And that is one of the many ways that they are able to suppress these suppress pathogenic bacteria. So while I didn't control it specifically, I did have in mind that it would be one of the many factors that helped inhibit and degrade biofilms.
Yep. And I have a question about, so in a lot of those graphs, you saw either where the mixed culture was protecting the biofilms, or in fact the single culture of lactobacillus that was degrading them. What do you think the main difference is between the mixed cultures of the probiotic versus the single cultures? Where do you think that mechanism is?
I definitely think it's with the multiple species probiotic, more synergistic properties were definitely expected. So multi-species probiotics are definitely, obviously, more commonly seen in nature and natural microbiota. And it's these communities that are usually able to work together and be able to fend off harmful organisms. So it was expected that the multi-species was able to inhibit or degrade better, which is definitely seen in the inhibition trials, where they are able to inhibit by 70%, while the single species only inhibited by 20%. However, this was definitely the opposite in the degradation trial. And yes, which was quite an interesting occurrence. And there probably are quite a lot of factors involved, including what else might have been present within the culture supernatant, as well as the different properties of the lactobacillus species themselves.
Yeah, thank you very much for that. I'll open it up for any last questions. Oh, awesome. Thank you very much, Cece.
Thank you.
So we've had our 4 finalists for the Australian Stockholm Junior Water Prize today, and I'm just blown away once again by the quality of the work that these students are showing us in the projects and the outcomes that they've delivered. And I think one of the highlights is that we've got a lot of work to do in some of these areas, and some great advice is where we should be taking these projects. And I hope that all of the finalists do stay connected with the water industry, no matter what your future holds. And hopefully, Miriam, you get a chance to go back and do some more projects with Hunter Water. I'd really like to I think we're going to announce the winner really soon. And I hadn't changed my page, so I hadn't known until just that moment. So without further ado, I would like to announce the winner. The winner of the 2022 is Julia Cummins. It's really hard when there's lots of clapping involved. So, but congratulations, Julia. Julia will be presenting the award to you in person at the gala dinner at Ozwater. I think you've got to get in quick for your tickets.
Okay, thank you.
I'm really pleased. Julia, do you want to say a few words?
Thank you. I'm incredibly honoured to have won this prize. Thank you so much, my science teachers. I know that they're watching the presentation, so thank you so much for helping me the past year and providing encouragement and support. I really appreciate it, and it's given me so many wonderful opportunities, and I'm really excited for the future and to present at Ozwater. Thank you everyone for coming today.
Thank you so much, Julia. And as Tomo mentioned earlier today, Julia will now represent Australia on the international stage and compete against 35 countries later this year. And I hope that you'll get the opportunity to actually travel to Stockholm.
Yes, that would be amazing.
Just wear a mask from now on. So I'll hand over to Tomo.
Thank you, Sandra, and congratulations, Julia. Well done. And I'm very hopeful that I get to travel to Stockholm with you. Okay, so I'll be in touch. So I'd like to also say a final thank you to all our finalists today. You've each got bright futures ahead of you, and we look forward to hearing more from you soon. Thank you also to our sponsor Xylem, and to each of you, our audience, for joining today's webinar. We hope you've enjoyed it. I wanted to quickly mention that the Australian Stockholm Junior Water Prize 2023 is now open for entries. Please visit our website for more information, or please do not hesitate to contact me directly if you have any questions. Thank you and have a good day.